Scientists have been able to send rats to sleep by dialing down the activity of a central region of their brains, similar to turning down the volume on a radio. By tweaking the activity of cells in a region called the central thalamus, the researchers found they were able to effectively control the consciousness of the animals. Researchers said that this part of the brain can be manipulated almost like a dial, to turn up the animal's state of arousal, or to send them to the land of Nod. The findings are published in the online journal eLife. The thalamus is region found deep in the brain which acts as a sensory hub, relaying signals from different regions and regulating consciousness and arousal. Playing with consciousness. To test the link between the activity of the central thalamus and consciousness, a team at Stanford University used fiber optic wires to stimulate the brains of anesthetist rats. Brief laser flashes were used to stimulate neurons in the central thalamus as the animals dosed. The researchers found that increasing the frequency of the flashes, to 40 to 100 pulses per second, woke up the rats. But dialing down the frequency to around 10 pulses a second caused the rodents to stiffen and stare in a seizure-like state before they returned to sleep. Functional MRI scans of the animals showed that, depending on the frequency of the laser pulses, different areas of their brains lit up. Those areas found to be more active during high-frequency stimulation when the animals were awake, were found to be inhibited at lower frequencies, such as when they were asleep. The brain scans showed the interaction between the different regions of the brain, with increased activity in one leading to a drop-off in another. Neurons in the somatosensory cortex, surrounding the central thalamus, become more active when the animals' brains were more stimulated. According to the researchers, their findings demonstrate that the cells in one region in particular, the zona incerta, fire in unique spindle patterns during states of low activity. These spindle patterns are seen in sleep, but not during high-frequency stimulation. Commenting on the findings, Yejun, Janet, program director at the National Institutes of Health's National Institute of Neurological Disorders and Stroke, NINDS, said, "This study takes a big step towards understanding the brain circuitry that controls sleep and arousal. We showed how the circuits of the brain can regulate arousal states," explained Dr. Lee. "We hope to use this knowledge to develop better treatments for brain injuries and other neurological disorders." Neuroscientists have long pointed to the idea of a sleep thermostat inside our brains which controls our conscious state. Last year, researcher from the University of Oxford experimenting with fruit flies found that our brains may even have a sleep switch. The activity of groups neurons in the brain may build until it reaches a critical point, calling for the brain to go offline. While scientists are still picking apart exactly how these processes are regulated, the findings could ultimately help people with debilitating sleep conditions, such as chronic insomnia.